sorry. What is the function of enzyme and how enzyme is actually playing a role in metabolism as a whole. Okay, all right. So when we talk about enzymes, so generally enzymes are biological catalysts that speed up the rate of biochemical reaction. Okay, when we said that enzymes are biological catalysts, meaning that enzymes would speed up all the process, especially in terms of the biochemical reaction. Okay, so I have a question. Will biochemical reaction still takes place without enzyme? What do you think?
Yes, madam.
Okay, why is that so? Why would biochemical reaction still take place?
Enzyme just speed up the whole process, but without other help, it's still working.
Yes,
in the body.
So very good. Okay, so enzymes are biological catalysts, meaning that biochemical reaction can still take place, but it will be at a much slower rate. So we really require enzymes so that the reactions could occur at a higher speed. Okay, so in terms of the structure, normally enzymes are three-dimensional globular proteins. So they are in tertiary and also quaternary structure. And some special RNA species can also act as enzyme, meaning that some RNA species can act as a biological catalyst that speeds up reaction. For example, ribozymes. Okay. So this is what we mean when we say that enzymes are biological catalysts. So what will enzymes do is enzyme will lower the activation energy. Okay. So how do we understand this graph? This is the y-axis is the energy required for a biochemical reaction to take place. Oh, before that, I have uploaded these slides in the Microsoft Teams. Okay, you can go under material. You can go at the general section and then go to files and then class materials. Okay, and then download from there. Okay, Sarumati. Okay. All right, so again, energy, the y-axis is energy, meaning that the energy that is required for a biochemical reaction to take place. Okay, and the x-axis would be progress of reaction. Okay, so in order from a reactant to be converted into a product, so a reactant is the starting point, the product is the end point. So in order for a reactant to be converted into the product, it has to go through the activation state. Okay, it needs to be activated. Okay, so usually without enzyme, the activation energy would be really high. Okay, so what enzyme does, it will lower the activation energy so that it will be more easier for reactants to be converted into products. Okay, so if without enzyme, then our body would take some time to gain the energy, okay, the activation energy to reach its transition state and finally be converted into product. So enzyme would lower that so that it would be faster for the reactants to become product. Okay, let's take an example. Let's take a reactant, for example, carbohydrate. Okay, so the product would be glucose that can be absorbed by our cells inside our body. Okay, so without enzyme, so this carbohydrate would take some time to build up or gain energy so that it will reach the transition state and be converted into glucose. So for example, in condition that we need instant energy, like we are running, or we are doing some intense exercise, so we need a lot of glucose in a fast, in a quicker time. Okay, so without enzyme, then by the time carbohydrate be converted into glucose, maybe we have already fainted, okay, because of the time taken. So with the presence of enzyme that converts carbohydrate into glucose, for example, that reaction could be speed up. All right. Okay, so again, enzyme would lowers the activation barrier and increase the reaction rate. Okay, but it does not affect the equilibrium rate. So the presence of enzyme would not mean that there's a higher rate of higher amount of product. Okay, the amount of product form would be still the same, but it will just increase the reaction rate, meaning that it will shorten the time taken for a reactant to become product, but it will not affect the concentration or the amount of products formed. Okay, it will just speed up the reaction. All right. So enzyme provide an alternative energy pathway that has lower activation rate than an uncatalyzed reaction. Okay, so the activation energy, or in short form EA, so this activation energy, short form is EA, is the energy required for reactants to reach the transition state where the bonds will be broken and new bonds are formed. Okay, so this is the transition state. So from reactant to transition state, the energy required is the activation energy, or EA. Okay, so that is just the definition. Okay, so let's look into the structure of enzyme. So what does enzyme makes up of? So in enzyme, we have active site. Okay, so if you could imagine the enzyme is something like in this form. Okay, so it has active site where the region is for substrate to bind. So let's say we have over here. So this is the active site. Kenapa ni? So let's say this one is the active site where the substrate will bind. Okay. Okay, and then we also have within the active site is where cofactors will bind and other prosthetic groups like ATP. Okay, for that reactants to become a product. Okay, so this active site generally occupy less than 5% of the total surface area, so it's very small compared to the whole structure of enzyme. And this active site, what's important for us to understand that it has a specific shape. Okay, specific shape meaning that not all enzyme can bind to any substrate or some random substrate. Okay, so each substrate or the reactants have their own specific enzyme to bind to. Okay, and a change in the shape of protein of this enzyme can affect the shape of active site and function of enzyme as a whole. Okay, so let's look into this in further detail. Okay, so this active site, they can be divided into two. So let me draw it again for reference. So let's say this is an enzyme. So let's say this is the active site. So this area is the active site. So within this active site, usually we have two sites. The binding site, let's say over here, and also the catalytic site. Okay, so within one whole active site, we have binding site and also catalytic site. So binding site is where the substrate will bind. Substrate or the reactants or the initial product. Okay, the first thing, for example, just now carbohydrate. Okay, so the catalytic site is where the catalytic action of enzyme presence takes place. Okay, there's another site of catalytic site within the active site. All right. Okay, so that's the structure of enzyme. We will look into details later. In terms of the mechanism of enzyme. Okay. Okay, so briefly, the names of enzymes usually, how do we identify enzymes are those that ends with ASE, like lipase, dehydrogenase, okay, transferase. So ASE, those ASE are usually enzyme. So usually the enzyme's name relates to the reactants that it binds to. For example, lipase. So lipase will catalyze the reaction of lipids, for example. Okay. When we have, for example, aminotransferase. So we could get some idea, okay, aminotransferase meaning that that's enzyme that's maybe transferring amino group, okay, something like that. So even, for example, sucrase. So the enzyme is sucrase. So the reactant or the initial product would be sucrose. Okay. So anything that ends with ASE is usually enzyme. And sometimes the name of enzyme can also describe the function of enzyme. All right. For example, oxidase. So if an enzyme name is oxidase, it could give us some overview that the enzyme could be catalyzing oxidation because of this oxide name in the oxidase. Okay, same goes to hydrolase. So this could give us an overview. Okay, this enzyme could catalyze hydrolysis because of the hydro word over here. Okay, but there are also some enzymes that are have different names. For example, the digestion enzyme ends with IN, like pepsin and trypsin. Okay, the name of enzymes is actually due to some historical situations. Okay, when for example, I can't remember, in 19 something. Okay, scientists have decided, okay, let's standardize the names of enzyme with ASE. So the enzyme that has been founded before that doesn't follow this rule. So that's why some enzymes may not end with ASE. Okay. All right. So this is the classification of enzyme. Just now I've mentioned that, okay, some enzymes are named based on its function. Okay, so these are some of the example of enzyme classes. So we have oxidoreductase. We also have transferase, hydrolase, lysase, isomerase, and ligase. Okay, so let's look into this in detail. So the first class of enzyme would be oxidoreductase, right? So when we talk, okay, oxidoreductase, meaning that it could be involving in oxidation and reduction. Okay, so that's why involved in redox reaction. So if you remember, this is just a recap. So oxidation is all about gaining of oxygen. Okay, oxidation, we give oxygen to that molecule. Okay, and then there's also loss of hydrogen, loss of electron, and increase in oxidation number. Okay, for example, Mg plus to Mg2 plus. Okay, things like that. And then we also have reduction. So the opposite of oxidation will be reduction. So vice versa of it. So if in oxidation we gain oxygen, so in reduction is about losing oxygen. Okay. Then we have gain of hydrogen, gain of electrons, and also decrease in oxidation number. Okay, so that is redox reaction. And the enzyme that's catalyzing redox reaction, we call them as oxidoreductase. Okay, so in this oxidoreductase, that's a general name, that's a general classification. Under oxidoreductase, we also have a more specific enzymes. For example, over here we have dehydrogenase. Okay, so the function of dehydrogenase, so how do we have a guess of it? So dehydrogenase, so it must have some relation to hydrogen. So what does it do to this hydrogen? It will dehydrogen it, meaning that this de meaning to remove it. Okay, so the enzyme dehydrogenase means the enzyme that's catalyzed the removal of hydrogen. Okay, and then we also have oxidase specifically. So in enzyme oxidase, what will this enzyme do? It will catalyze the reaction of adding oxygen to hydrogen and forming water. Okay, so gain of oxygen. Okay, so that's another example of it. So that is for oxidoreductase. Next one, the next group, we have transferase. Okay, so this one is quite straightforward. So when we say transferase, meaning this enzyme could be catalyzing process that transfer something. Okay, so they transfer a group of atoms from one molecule to another. For example, AX. So AX is one group, one molecule. And then we have another molecule B, or another reactant. So a reactant A, reactant 1, reactant 2, and then we have BX, the product 1 and product 2. Okay, so what this enzyme do, if you observe in this reaction alone, it's transferring this X group from A to B. So initially X is bound to A, but after the reaction, now X is bound to B. Okay, so this is the example. So we have, this is the reactant, the initial product. okay, aspartate and also alpha ketoglutarate. So this one is the reaction that's happened in protein metabolism, okay. So this is reactant 1, initial product 1, initial product 2, aspartate and alpha ketoglutarate. So PLP is a type of enzyme. Okay, PLP is actually a type of aminotransferase. So bawah or under transferase, we also have aminotransferase. So one type of aminotransferase is actually PLP. PLP. All right. So if you notice what this PLP do, it will switch or transfer between NH3 plus over here in aspartate and also the C double bond O in alpha ketoglutarate. So it will switch places between these two groups. And now we have oxaloacetate and also glutamate. So how oxaloacetate is formed is when this C double bond O from alpha ketoglutarate is transferred to aspartate and replace the NH3. So when where will this NH3 go? It will go to the alpha ketoglutarate and form glutamate. So if you notice, there's a switch here between NH3 and also C double bond O. So when there's a switch in place, now new product is formed. Okay, so that is the function of transferase. Okay. All right. So we also have transaminase, okay, transfer of amino group, and also phosphotransferase, transfer of phosphate group. Okay. All right. So the next enzyme we have is hydrolase. So hydrolase meaning that it can catalyze the hydrolysis of substrate by addition of water. Okay, so some reaction requires water molecule to be added to the reactant or the initial product. So this enzyme will speed up the addition of this water molecule to the product. Right? So that's what hydrolases do. It will catalyze hydrolysis. So hydrolysis meaning addition of water to the initial product. So AX plus water, now we have XOH plus HA, for example. Okay, so example of hydrolase is maltase. So what maltase will do, it will add water to maltose so that this maltose can be broken to glucose. Okay, and then we have another example, lipase. So when we say lipase is a type of hydrolase, lipase will cause the addition of water to lipid so that the lipid can be broken down into fatty acid and glycerol. Okay, so that is for hydrolase. Okay, the next example we have lyase. Okay, so when we said we will see this a lot. Okay, the term lyse. So to lyse simply means to break or to split. Okay. So this lyase will break the chemical bonds without adding water. Okay. Just now, in hydrolase, it will still break the molecule. For example, the lipase over here, it will break lipid into fatty acid. But hydrolases or this lipase require addition of water. Okay, but for lyase, they can break the bond without addition of water, without the help of water. So for example, directly, the initial product is A and B. So it will turn into A, B, for example, and separate it. Okay. So for example of lyase, the example of what lyase is decarboxylase. So the same goes to dehydrogenase. So if dehydrogenase meaning the enzyme that catalyzes the removal of hydrogen, so D means remove. So this decarboxylases meaning that it will remove carboxyl group. So D means remove, carboxyl is the group. Okay, so decarboxylase will catalyze the removal of a carboxyl group from an enzyme, from a molecule. Okay, for example, decarboxylase will remove the carboxyl group from respiratory substrates to release carbon dioxide. Okay, the C double bond O will be remove, for example, from a group, a molecule over here, so that CO2 can be released. Okay. So that is for lyase. Next, we have isomerase, isomerases. So isomers simply means, so in basic chemistry, when we say a molecule is isomer to each other, let me draw it. So let's say we have molecule 1 and molecule 2. Okay, when we say molecule 1 and molecule 2 are isomer to each other, or we say molecule 2 is an isomer of molecule 1, molecule 1 is an isomer of molecule 2, simply means that the molecule has the same structure, okay, same chemical formula, but it may have different arrangement, right? Okay, so for example here, isomerases will catalyze the conversion of one isomer to the another isomer by transferring a group of atoms from one molecule to another. Okay, for example, from molecule 1, we want to convert molecule 1 to molecule 2. Okay, this molecule has similar formula, but it has a different arrangement. So what will isomerase do? Catalyze the reaction from one isomer to another. Okay, so this is one example over here. Okay, if you can observe, so we have the initial, let's name this molecule 1 for convenience. Okay, so this is molecule 1, this is molecule 2. So if we write it in the chemical formula form, it will looks the same thing. It has the same number of carbon, same number of oxygen, same number of hydrogen, same number of phosphate, but it has different arrangement. Okay, so from molecule 1, we want to convert it to its isomer, molecule 2. That's when this triose phosphate isomerase plays its role. Okay, so this is the arrangement for molecule 1 in detail. So we have the CH2OH group over here. Okay, C bounded to 2 hydrogen and also the OH group. Okay, and then we have the C double bond O. Okay, C double bond O, and then the CH2OP. C bound to 2 hydrogen and also oxygen and phosphate group. Okay, now converting from this molecule 1 to this molecule 2, you will see that this isomerase will change the position of these 2 hydrogen. Okay, now this 2 hydrogen is actually transferred to the C over here. So that's why they have like this. Okay. Okay, so this is for isomerase. So simply means to catalyze the conversion of one isomer to another. And isomer means they have the same chemical formula, but different arrangement. Okay. Okay, so that is for isomerase. Now we also have ligase. Okay, if ligase means to break or to split. Okay, ligase means to glue it together. Okay, so ligase will catalyze the synthesis of new chemical bonds, and in ligases, usually the reaction requires ATP. Okay, for example, we have over here 2 separated or broken DNA strands. Okay, we have DNA strand A, DNA strand B. So this area over here is broken. So this ligase will come and fix the broken area and causes these 2 strands A, B to be ligated together or combined together, attached back and glued back together. Okay, so this reaction of combining DNA strand A and DNA strand B is utilizing ATP, requires energy. Alright, so lyase means to break, so ligase means to join back. Okay, synthesis new chemical bonds. Okay, between these 2 molecules, for example. Okay, so we have covered the types of enzyme just now, or classification of enzyme. So next, let's look into where are these enzymes located, or the localization of enzymes? Okay, so it can be further divided into two, the intracellular and also extracellular. Okay, intracellular means inside the cell. So many enzymes are localized in specific organelles within the cell. So if you remember in chapter 2, you did cellular biochemistry, all these organelles and also structures that you label inside a cell. So we have one cell, then we have nucleus, for example. So this is nucleus. And then within a cell, we have many other organelles. For example, mitochondria, we also have lysosome, and so on. Okay, so in this, each organelle, they may have their own enzyme to catalyze a certain reaction. Okay, for example, in lysosome, so lysosome contains also enzyme like lysozyme. So in lysosome, so the function of lysosome is to break down or degrade a complex macromolecules. So it requires enzyme to do so. So enzyme in lysosome will facilitate the degradation of complex macromolecules. Okay, same goes to nucleus. So just now, as I mentioned in this slide, so we have ligase that, for example, DNA ligase that synthesis back chemical bond between two broken DNA. So this DNA ligase is actually in the nucleus. Okay, so it will repair the broken DNA inside the nucleus to synthesis a new one, okay, or repair the broken ones. Okay, and we also have within the cytosol itself, meaning cytosol meaning this area outside of organelle but still inside the cell over here. So this is all the cytosol area. Also have enzymes, okay, for the process of glycolysis or fatty acid synthesis or urea cycle. Okay, we also have enzymes in mitochondria, which we require them for Krebs cycle or TCA cycle. So TCA cycle mainly for carbohydrate metabolism. We also have fatty acid oxidation. How do we utilize the fats that we have eaten? And also urea cycle. So urea cycle is just mainly for removal of urea or toxic waste from our body. So all those enzymes are contained in mitochondria. Okay. So although we observe that organelles has already been like the smallest thing that we can observe in our body, so within all those organelles, we still have a lot of other things, for example, enzymes, okay, to facilitate the biochemical reaction in it. Okay, that's for intracellular. There's also enzymes that are being secreted out of the cell. Okay, so for example, when we have cell, so these enzymes are being secreted out of the cell to facilitate the biochemical reaction that needs to occur outside of the cell. Okay, so so those enzymes, although being produced inside the cell, but it will perform its function outside of the cell. Okay, for example, the digestive enzyme. Okay, so if you could imagine, okay, this is the stomach. Okay, excuse my drawing. So this is the stomach. So this is the stomach lining. Okay, within the stomach lining is actually epithelial cells. Okay, we have cells lining the stomach. Okay, so what will these cells do? They will secrete enzyme inside the stomach area. Okay, so enzymes are coming from these epithelial cells that lining the stomach. So whenever food that comes in, so this enzyme will break down the food. So that's example of extracellular enzyme, meaning that it will take place outside of the epithelial cells inside the stomach area. Okay, and for other examples could be amylase, okay, secreted by the salivary glands, but the enzyme would perform its reaction within our mouth cavity. Okay, and other enzymes. Alright, so that is for localization of enzymes. Okay, before we proceed with enzyme shape and reactivity, can we take a 2 minutes break? I will end the Zoom session and then come back at, okay, let's take a 3 minutes break, 11:45. Okay, come back using the same link.
Okay,
Alright,
madam.
see you later.